Hi, Miss Nikki here. Welcome to Chapter 29, Development, Part 2. We really kind of talked about all of this in the first part. We talked about this pre-embryonic period. We talked about gastrulation and the endoderm, ectoderm, mesoderm. We're kind of here looking at organogenesis, how basically the framework is laid down, and then we're building on those organ structures. We're really going to start going through some of the hormones that are released. And we'll talk about labor and delivery, and then at the very end we'll talk a little bit more about some of the genetics. Here we have hormonal changes with estrogen and progesterone. We've been talking about these two for quite a while now, right? The corpus luteum during the first trimester is producing the majority of estrogen and progesterone. The placenta will take over in the second and third trimesters. And you're going to have a high level of suppression of FSH and LH, right? So we do not want any more follicular development. So during pregnancy, there's no more secondary oocyte ovulation. We're going to see that estrogen and progesterone are going to cause the uterus to become larger. Mammary glands are going to become larger. The fetus is going to grow. In females, you're going to see nails growing, fuller hair. We're also going to see the relaxation of ligamentous joints, specifically in the coxal bones, right? We're going to see the functional layers of the uterus are going to grow due to the progesterone, so that would be that endometrial layer. We can see this hormone here, relaxin. It's going to be secreted by the corpus luteum and the placenta. It's going to promote blood vessel growth in the uterus. Obviously, the fetal chorionic villi are sitting in pools of mom's blood, and we need to make sure that we're exchanging nutrients properly, so you can see why that would be important. We also have this corticotropin releasing hormone. This is secreted from the placenta. It's going to have something to do with the timing of childbirth and also responsible for an increase in aldosterone in the mother. So remember, aldosterone reabsorbs sodium, which causes water to be reabsorbed as well. So we're going to see fluid retention and edema in the mother, especially towards the third trimester. There's also this human placental lactogen, and although this makes you think it has something to do with milk, in pregnant females, humans, we see this secreted from the placenta, and it really has more to do with metabolizing certain nutrients. So mom is going to use more fatty acids instead of glucose and save that glucose for the fetus, but also remember, glucose is what your brain wants, right? Your brain, your central nervous system wants glucose, doesn't want to turn fats into ATP energy. We also have prolactin, so this does have something to do with ensuring milk production, right? And it's increased about 10 times closer we get to delivering. We also have oxytocin. Remember, this is that milk letdown from the mammary glands, so you need prolactin to make the milk and then oxytocin to release the milk. It's also involved in uterine contractions. You can see how that would be useful for childbirth, right? And the increase in oxytocin happens in the second and third trimesters in response to the rising estrogen levels produced by the placenta. Just go back and make sure you remember that oxytocin is stored in the posterior pituitary, but it is actually produced or made in the hypothalamus. I put this picture in because I think it's interesting to see kind of before conception. You can see the uterus here, and the uterus is growing. It's coming all the way up to the xiphoid process when we're in the final stages of fetal development. So we're going to see some changes in mom's digestive system, nutrient uptake, some metabolic differences that are happening in mom. So we're going to see kind of this morning sickness, probably due to those high levels of estrogen and progesterone and all these new hormones that mom is making, right? We're going to see increased insulin resistance. So remember, insulin kind of helps cells take up glucose, right? Cell uptake. Well, we said we're trying to conserve glucose for the brain and for the fetus, and we're trying to burn more fat, so it can cause some issues with insulin resistance. We're also going to see intestinal mobility slow down. We're going to see constipation. So again, think as that uterus is growing all the way up to the xiphoid process, and then it's also pushing down weight, right, on the bladder and the intestines. intestines, get it out. So you can imagine that that's going to put pressure on your large intestine, that could put pressure on your small intestines and affect the way that food moves through them. You can also imagine the upward push is pushing on your stomach and pushing it into your diaphragm. It could make digestion hard. It could make breathing hard. For nutrition, mom has to have an adequate supply of folic acid, which helps with brain and spinal cord development. Hopefully you're thinking calcium and bones, right? Proteins. So mom has to eat proteins so they can be broken down to amino acids so that we can make whatever fetal proteins we're making. Remember all that connective tissue that we're making? We got to have elastic fibers, we've got to make muscle, we've got to have myosin proteins. So lots of fetal proteins have to be made. Mom has to consume enough protein in order to supply those amino acids to make those fetal proteins. And then the iron, hopefully we're thinking of that heme group inside the red blood cell. I mention this because it is something pretty common. Diabetes that first develops during pregnancy gives you an increased risk of having type 2 diabetes later in life. So usually they're going to put you on some sort of special diet to regulate your glucose. It does usually cause kind of a larger baby, so increased chance of having to have a cesarean section instead of having a vaginal delivery. Again, changes in mom's cardiovascular and respiratory. You're going to have increased plasma volume. You're going to have increased cardiac output. That means your heart rate's gone up, your stroke volume's gone up. You're going to see compression of those abdominal blood vessels because the fetus is pressing down on them. It might impair venous return from lower legs, right? So you're changing the pressure differences, right? And we said pressure differences is what allows the exchange of nutrients and fluid, right? And then you're also, we just talked about aldosterone, right? So you have increased aldosterone, so you have increased swelling or edema. And then if we're pressing on the abdominal blood vessels, that as you breathe in really deeply, it's supposed to move that venous fluid from lower limbs to the abdominal cavity. That might cause some issues, right? So prevents the diaphragm from fully distending or expanding, can affect your respiratory rate, your oxygen consumption rate, your urinary system. It is having to filter more blood, right? Not only yours, but the fetus. And then sometimes the ureters or ureters can be compressed by the uterus. So preeclampsia is a condition where high blood pressure is occurring. It usually happens in the later stages of pregnancy. Risk factors, obese, diabetes, older in age. Causes unknown, general risk of hypertension for the mother. There's some sort of poor perfusion going on between the placenta and the mom. The only cure is to give birth, right? So they might put you on bed rest or give you medications if they're trying to hold off on giving birth. If it's maybe a little bit too early and they want to just wait a little bit longer. So eclampsia, high blood pressure can cause seizures. This is a medical emergency. Labor, the technical definition here is expelling the fetus, right? Expulsion of the fetus and the placenta from the uterus, usually at about 38 weeks. Not all uterine contractions are true labor. So sometimes the labor is what we would call false labor, and there's a slide coming up. So contractions are usually weak, they're irregular. Premature labor is anytime before that 38 weeks. We're not really sure that the lungs are fully developed. We'll talk about this when we get to the respiratory system, but the fetus has to be producing enough surfactant, and as long as their lungs are fully developed, then premature labor is not terrible, right? So it says undesirable, but we know plenty of people who've been born at 35 weeks, 36 weeks, and the baby was fine. False labor is when we have uterine contractions that do not result in the stages of labor. So they're usually irregularly spaced, they don't become more frequent, they're weak. This is usually the hallmark if the pain stops with movement. If you turn to your side and the pain stops, that maybe it is this Braxton Hicks contractions, which is false labor. And basically this false labor, it does not lead to cervical changes. So we're going to see on the next slide that the cervix is going to thin and dilate, right? Effacement and dilation. So these false labors, these Braxton Hicks contractions are not enough to cause changes in the cervix. And we'll see that this is due to secretion of hormones. So here we have true labor, uterine contractions that increase in intensity and result in changes to the cervix. So not only is the mom's hypothalamus releasing oxytocin, but the fetus hypothalamus is also secreting oxytocin. These stimulate the placenta to secrete prostaglandins, and these prostaglandins are going to stimulate the uterine muscle contraction and soften and dilate the cervix. So true labor has to be a combination or enough oxytocin is being secreted to initiate true labor, which results in changes to the cervix. And remember, if for some reason the true labor is not happening, if you're not producing enough oxytocin, we can induce labor by giving you Pitocin. So positive feedback mechanism, I'll show you the picture in just a second. This is kind of the wording. So as the fetus's head is pressing on the cervix and causing the dilation of the cervix, it causes the hypothalamus to secrete more oxytocin, then the placenta secretes more prostaglandins, then we have more intense uterine contractions. This is going to continue until the fetus is expelled, right? So once the fetus is expelled and the placenta with it, the prostaglandins are removed, right? Because they were secreted from the placenta. The uterus and the cervix are no longer fully stretched. We're going to see a drop in oxytocin and labor ceases. Nice image here. You can see stimulus, oxytocin from the fetus and the mother, stimulates uterine contraction. Uterine contraction causes the head of the fetus to press against the cervix, making it dilate, and that stimulates the hypothalamus to release more oxytocin. So that's that positive feedback mechanism. We also know that the oxytocin released stimulates the placenta to make prostaglandins, and then those cause more concentrated contractions in the uterus and kind of adds to this positive feedback loop. Dilation stage, this is the first stage of labor. This is where we're going to see the thinning or effacement or effaced and dilated cervix. The amniotic sac is going to rupture and that amniotic fluid is going to be released. We can see here, hopefully you can see the edges of the cervix. So the cervix has started to dilate. You can see that there's compression here on the bladder. When we get to the final dilation stage, you can see that the parts of the cervix are pressed up against the uterine wall. So now we have a fully dilated cervix. Also notice what's happening to the bladder and notice what's happening to the rectum. So it's very common during childbirth, the next couple of pictures as the baby is being kicked out of the womb, right? It's very common for women to urinate or defecate during the labor process. So the expulsion stage, we see the complete dilation of the cervix. This can last several hours. The uterine contractions are going to help push the fetus through the vagina. We're going to see a crowning event when the first part of the baby's skull, the clavarium, is in the vagina. And then sometimes an episiotomy is necessary to widen the vaginal opening. So you can see here now, barely see the bladder. You can definitely see how the rectum is being compressed here. You can also see the crowning of the baby's head in this image, and an episiotomy would happen here if we needed to make this opening a little bit wider. You can also see that the baby's face is facing downward, right? So here's the expulsion and the crowning phase. Episiotomies aren't really done anymore. I mean, maybe in some cases they are. I'm not an OBGYN, but they've really stopped doing them as much as they used to, because if you cut with a blade, you might be cutting across some collagen in the dermis, and that makes scar tissue develop. It's harder for it to heal. So now in some cases they think kind of naturally tearing is probably better for healing than to have a surgical cut of episiotomy. In the placental stage, after the baby is expelled, the uterus has to contract again because you have to displace the placenta from the uterine wall. So we do not want to leave these fetal membranes, right? They're not part of your immune system. If these fragments are left, we can have complications, bacterial infections, sepsis, things of that nature. So here we can see the umbilical cord, we can see the placentas detaching from the inside of the uterus, and then they give you kind of a nice picture here of what the placenta would look like. So we do have vaginal delivery versus C-section or cesarean delivery. So vaginal, born through the vaginal canal. C-section, they're going to cut through your abdominal muscle, through the uterus from the outside, and pull the baby out that way. So one of the things that's limiting for a C-section is the baby's not exposed to the fecal bacteria during a vaginal delivery. So we want, remember how the baby's face was pointing down, right? So the baby, as it's coming out of the vagina, it's exposed to all of mom's bacteria, right? All of mom's gut microbes. So these are diverse and individual people, right? Protective, and they're going to have nutrients that are found in breast milk, but you are getting the first dose of what your gut bacteria are going to look like from a vaginal delivery. So exposure to surrounding environment during a C-section, maybe not as good. You're not getting as diverse a population, right? Prone to health issues later on in life, more susceptible to hospital-acquired bacteria. Postpartum, this is the time period after giving birth. We have further changes, right? We have to feed the neonate, so we have to produce milk. We have to have the letdown of that milk from the mammary glands. And then our uterus has to return to that pre-pregnancy form and function. We're also going to see estrogen and progesterone levels plummet. Remember, the placenta was delivering the majority of those hormones, and so now the placenta is gone. So sometimes this can trigger depression, where you hear people talk about postpartum depression or baby blues, right? For lactation, we need to make breast milk from the mammary glands. And we all know that prolactin is responsible for milk production. It is produced by the anterior pituitary. And we talked previously about dopamine, secretion of dopamine being inhibited. We talked about prolactin inhibiting hormone, that is dopamine. Before, they used to think there was a prolactin releasing hormone. So high levels of estrogen are going to cause an increase in prolactin being produced by the anterior pituitary, and it's going to trigger milk production. Colostrum is produced by the mammary glands in the first few days after birth. This is a watery milk-like substance, but it has lower fat than true breast milk, and it has lots of antibodies, lots of immunoglobulins. So this is a way for the infant to acquire that passive immunity from the mom. So the infant hasn't been exposed to these pathogens to produce antibodies, but mom has. And so mom is passing those on in larger quantities within the first couple of days via breast milk. There's also a laxative effect with breast milk, and that helps facilitate the first bowel movements in the infant. So milk letdown, we all know, is because of oxytocin. So this involves a positive feedback mechanism with suckling. Mechanoreceptors in the breast are stimulated, sends a message to the hypothalamus, and causes oxytocin to be released from the posterior pituitary. Well, let's follow one pathway and then the other. We can see stimulus here, right? So the infant is suckling. That's causing a message to be sent to the hypothalamus. The hypothalamus is going to tell oxytocin to be released from the posterior pituitary. That's going to stimulate the mammary glands to contract, and that's going to release breast milk. So this keeps going, right? So oxytocin allows the milk to be released from the mammary gland as long as the infant is stimulating the nipple. So the suckling also inhibits dopamine. So that allows for the anterior pituitary to secrete prolactin, which targets the mammary glands and produces the milk. So they're both kind of acting at the same time. Not only do you have the infant suckling that tells the hypothalamus to release the oxytocin to release or let the milk down from the mammary glands, you also are controlling the production. So the infant suckling, you're going to release what you have, but you're also going to stimulate the mammary glands to produce more. Now we get to move into the human genetics. I'm just going to go over the definitions. There's very few questions about this. Some of the key terms would probably be the best thing to know. Like I have karyotype listed here, display of chromosomes, their pairs ordered, arranged by size, and there's an image coming up. We know about autosomes already. We know about genetics. We know homologous chromosomes, right? Paired chromosomes with genes for equivalent characteristics. We talked about sister chromatids, right? And we talked about homologous chromosomes. Here's our karyotype. Basically, they take a sample of cells that have DNA and they tell that cell to grow up and they initiate division. And as the cells are lining up to divide, then we arrest them. We have computer programs that will tag certain pieces of DNA and it'll fluoresce in different colors, like you can see here. And then the computer will sort them all out and take a picture. So you can see this individual right here. This is XY, so this is a male. You can see the 22 pairs of autosomes. And what you might be able to see is if somebody had trisomy, they might have an extra 21 right here. So what's the difference between a gene and an allele? So a gene codes for a protein, and allele is a variation of that. So if I have 2 chromosomes, and right here where the X is, that's a gene for hair color. An allele would be, could I have one gene for blonde hair, and could I have a second gene for brown hair? Maybe you got a blonde-haired allele from your mom and a brown-haired allele from your dad. So a gene is a unit of DNA that codes for a protein, and an allele is a variation of some gene. Dominant alleles are going to show, they're expressed, they're going to mask the recessive allele. So the only time you get the recessive is if it's expressed on both of the chromosomes. So if you have dominant, so dominant, or dominant and recessive, both of these, you're going to show, let's say, brown hair. The only way you're going to get blonde hair is if you have 2 recessives. And we've done Punnett squares before, basically a box showing the different gene combinations from the 2 parents. Basically, we're looking at offspring. So homozygous, identical alleles. Big A, little a, little a. Heterozygous, you have 1 dominant, 1 recessive. Genotype is your genetic makeup, phenotype is your physical. So we could do genotype XX. What is your phenotype? What do you look like? In most cases, right? Barring any unusual issues. Or you could say, we said AA was brown hair. So genetically, you're AA. What is your physical? You have brown hair. So phenotype, physical, genotype, what do your genetics say? And here's an example of a Punnett square. They're looking at widow's peak. So this individual has a widow's peak, right? So they're looking at the egg and sperm. So the male has 1 dominant and 1 recessive. The female has, this is female. The female has 1 dominant and 1 recessive. So these are the possible egg combinations, and this is the possible sperm. And then we can look, do a Punnett square and look at the offspring and the percentage of what the offspring could possibly have. So remember, when creating gametes, you're having the DNA, right? So you're going from a diploid to a haploid. So one of the eggs is going to get the dominant and one of the eggs is going to get the recessive. And we don't know which egg is going to be fertilized, just like we don't know which sperm is going to fertilize which oocyte. So these are all the possible combinations. You could get somebody that's homozygous dominant. You could get an offspring that is heterozygous dominant, another one that's heterozygous dominant, and then you could get one that is homozygous recessive. I put this slide in just so we can realize the degree of genetic variation that we have. So we've talked about the crossover of those homologous, those homologs, those chromosomes, those tetrads that we had, right? So remember in prophase 1, we had the crossing over of pieces of genetic information. The independent assortment is saying that we don't know how the chromosomes are going to line up, right? So remember, they're line up on the metaphase plate and then split. It's not like they have parking spots. It's not like chromosome 1, chromosome 2. They can be in any arrangement. They could be flipped. You could have dad on this side, mom on this side, right? So the chromosomes can be flipped several different ways, so they don't line up in order. So having independent assortment, having the crossover, and then the randomness of fertilization. We don't know which egg is going to be selected to grow up into the secondary oocyte. We don't know which sperm is actually going to make it to the egg to fertilize. So we have 1 out of 72 trillion zygote possibilities. That is a lot, right? So this is why we are all genetically unique. When I go through these patterns of inheritance, I'm just showing you that there are other options besides this dominant and recessive. So when we say dominant allele always expressed in the phenotype, there are relatively few traits that follow this pattern. So even though dominant and recessive are the easiest, the most straightforward, this doesn't happen that often. It's always more complex than we think. So incomplete dominance is when you have 2 heterozygous alleles and the phenotype is in between. They give you the example of sickle cell because we're talking about human anatomy, but before we look at that, think about if you took a white flowering plant and you took a red flowering plant, would you be totally surprised if you had a few plants in the next generation that were pink? Probably not, right? So this would be an example where the white doesn't completely mask the recessive red. So you have this kind of in between the 2 original parent phenotypes. So for sickle cell, most individuals have 2 identical alleles, and they're showing you a dominant here, coding for normal hemoglobin A. Sickle alleles produce abnormal hemoglobin. Sickle cell disease if 2 homozygous recessive alleles. If you're heterozygous, you're carrying the sickle cell trait. So you're going to have some issues. You're going to have oxygen saturation issues, right? But it's not as severe as having sickle cell disease. And they've labeled this a little bit differently. I'm going to say AA, this is normal. If you're 1 dominant, 1 recessive, so you're heterozygous, you're a carrier. And if you have 2 recessive, if you're homozygous recessive, then you're going to have sickle cell disease. Sickle cell disease. So this person may not be asymptomatic. This person may have some symptoms like low oxygen saturation, right? So this dominant doesn't completely mask this recessive. We also have codominant inheritance where the 2 alleles are equal. We saw this in blood grouping. You remember A and B? So we had our red blood cell. Did it make an A tag and did it make a B tag? They were both equally expressed. Polygenic inheritance is where we have multiple genes that are interacting to produce this physical phenotypic trait. So there are genes on different chromosomes, and most human traits are the result of this. So eye color, it's not straightforward. It's not one gene. Neither is height, neither is skin color. So expression of traits are also influenced by the environment. The very last slide that we'll look at, we'll talk a little bit about this, but if your genes, so I'm going to say if your genetic said you should be 6 foot tall, what happens if you don't have enough nutrition? What happens if you have low calcium? And remember, bones are built by calcium, right? So let's say you only make it to, I don't know, 5'8". This is what your genetics said, but this is your physical due to environmental factors. So this is a nice example of kind of that polygenic. So these are separate genes on separate chromosomes. And the skin color depends on how many dominant you have versus how many recessive you have. So here we have really light skin color, and here we have a darker skin color, and you can see the more dominant alleles you have, right? The more dominant alleles you have, the darker your skin tone is. So it's a combination of multiple genes on different chromosomes. We also have sex-linked inheritance. So traits are expressed on the X or the Y chromosome. Most of the Y is male development. Most of the times when we see a sex-linked trait, it involves the X chromosome. So X-linked recessive traits, it's always expressed in the male because only one bad copy or recessive copy is going to be available to the male. So what they're saying is if you're a female and you have a trait, recessive trait here, right, you have to have 2 copies for that recessive trait to show up. But if you're a male, you only have to have 1 copy and that trait's going to show up. So women can also be carriers. What if you had 1 normal and 1 recessive? Then you'd be a carrier of that trait. So females it's a little bit easier for them because they have a secondary X that could be producing the protein from the gene, so they don't see the effects as much. In hemophilia, this is a blood clotting disorder, so individuals are bleeding profusely after injury. Remember we talked about some of these, factor 7, factor 8, some of the ones that we mentioned in more detail when we did the blood chapter. Females are carriers, but the males are affected if they're carrying the gene for hemophilia. So again, here's the male with the recessive hemophiliac. They're missing this factor. They're going to be affected. Here we have a female. She has 1 normal, and she has 1 gene that's missing. So one of these factors is missing on this chromosome. Well, this normal can compensate for this other gene that's defective. There's a lot going on this slide, but we kind of mentioned this already. They want you to see that if the disease is carried on the X chromosome, that sons are more affected. If the female contributes her egg with the diseased allele, then the other allele came from dad, so she's just a carrier, right? She may or may not have symptoms for that X-linked disorder. It was kind of like what we said before, you're a carrier. You have 1 dominant and 1 recessive. On this case, you have 1 good allele and you have 1 bad allele. But with sons, they have like a 50-50 chance here of getting the diseased allele versus the normal allele because they don't have the Y chromosome bringing that extra X. If it's a son, it had to have a Y chromosome coming from the sperm. So bottom line, they're trying to say if you're a male and your mother is a carrier of an X-linked disorder, you have an increased chance of actually having the disease because you only have 1 X chromosome and you don't have a backup copy to mask out any issues that you might have or might be carried on that chromosome. This last slide is basically saying that your genotype doesn't always have to match your phenotype. So you could have this particular genotype and phenotype you should look or have disease symptoms X. But there are some people that have this genetic code and they have no symptoms, right? So they want you to realize that it's probably about 50-50, 50% genetics and 50% environment. So the environment can affect our genetic traits. They can influence those genetic traits. What type of chemicals are you exposed to as you were developing? And then give you an example in the book. They talk about fetal alcohol syndrome, where mom is consuming tons of alcohol and that it actually affects the development of the fetus. Poor nutrition can also have a negative effect on development if mom isn't eating properly while the fetus is developing. They want you to see that there are alleles that have a risk of cancer development, but you may never develop cancer. It just depends on those environmental factors, right? What's your nutrition like? Do you smoke? Do you work out? Do you have good cardiovascular health, right? So they want you to realize that your genetics are not the only factor. We also have to work or think about environmental factors on us while we're developing and then also as we're aging. Well, I hope you enjoyed this chapter. I'll talk to you guys again soon. Bye.